A very good evening to everyone. Welcome in the class. The topic of today's class is direct and indirect gene transfer methods. So specifically, when we are talking in terms of plants, we'll be dealing it that ways because in this series we are doing that how to make plants without the help of the normal fertilization and conventional method. We are seeing some different ways of preparation of plants, right? So that is the way we are dealing. So this direct and indirect gene transfer method is in context to the plants. So let us start this. And as we say in the starting, let's crack CSIR UGC NET together. Unacademy is one of the India's largest platform for the preparation of CSIR UGC NET. A little about myself. I'm your educator Farida Bandookwala, and I have a teaching experience of 10 years. I've qualified MP SLET examination, and presently I'm pursuing PhD from School of Biotechnology DAVV Indore. If you're opting for an academy, what all things you are going to get? You are going to get daily life classes. You're going to get the whole syllabus in a well structured manner, and there are 13 units. You are going to get live tests and quizzes, and if you're opting for plus subscription, you will have unlimited access towards all the videos and sessions recorded under the category of CSIR. If you're opting for the CSIR UGC NET, you'll be getting the videos related to GATE, JRF, and for all the top educators as well. Unacademy tries to give free live classes on YouTube, and it tries to give free live classes on special platform. So presently you're watching my free video on YouTube, and you can get the benefit of watching the YouTube videos just by subscribing the channel, so that you'll be getting the notifications of all the upcoming videos. And what about special classes? For special classes, you need to download Unacademy Learning app. When you will download the Unacademy Learning app, you will need to make your account, and then you can follow my profile by the name Farida Bandookwala, and you will be able to get all the videos whichever I have done previously, and you will get the notification of all the upcoming videos whichever I'll be doing. After that, Unacademy has got one more platform where it takes the lecture. So this is plus subscription, and it is a paid subscription, and recently a major discount is going on. For 12 months 54% discount is going on, and for 24 months 67% discount is going on. So what I would suggest is go for 12 months or 24 months for this, and it is quite pocket friendly amount, 11,500 for 12 months and 15,800 for 24 months, and you will be getting an additional 10% discount if you are using my referral code FB01. Do not forget to use my referral code FB01 to avail an extra 10% discount. So the amount will come down to 10,395 for 12 months and 15,120 for 24 months. Do not forget at all to use my referral code. And the prices are soon going to increase from 31st of December. So do not make any late and do not delay, because the sale is going to end. So just take your subscription, and if you're opting for uh, the plus subscription through my referral code, then also you'll be able to get the videos of all other educators who are teaching on the platform of Unacademy. So by one subscription you will be able to get the videos of all other faculty members. So let's crack CSIR UGC NET and GATE together in morning sessions. We do study for the GATE sessions, and in the evening session we study for CSIR. So the topic of today is direct and indirect gene transfer. So first of all, let us see gene transfer. Now when we are saying gene transfer, we are talking it in terms of plants, right? This gene transfer method in terms of plants, because we have seen firstly artificial seeds, then we have seen hybrids, then we have seen the protoplast fusion, we have seen hybrids, and in today's class we are going to see that how the genes can be transferred in plants. This is the topic of today. So the gene transfer technique in plants, in plant genetic transformation, are broadly grouped into two categories. So genetic transformation, genetic transformation can be done two ways. Those who are attending the class live, please they can tell me whether my voice is clear or not. So first is vector mediated method. Vector mediated method. This vector mediated method is also called as indirect method, and second, direct or vectorless method. Vectorless, and this is called as direct method. So these are the two ways through which genes can be transferred in plants, right? So first of all, we'll be talking about indirect method or vector mediated method. Please, those who are listening the class live, please can they tell me whether my voice is clear to them? Indirect method or vector mediated or vector mediated gene transfer. This is the thing that we have to see. Now, vector mediated gene transfer is carried out either by Agrobacterium tumefaciens or Agrobacterium mediated transformation, or through viruses. So indirect method, when we are talking, so it can be through two ways. First is it can be Agrobacterium mediated, Agrobacterium mediated. Secondly, it can be through viruses, plant viruses, specifically plant viruses, right? So first of all, we'll be talking about Agrobacterium mediated gene transfer. Agrobacterium mediated gene transfer. Am I audible? Am I clearly audible to you all? Please reply. Agrobacterium mediated gene transfer. So what is Agrobacterium tumefaciens? Agrobacterium tumefaciens. This Agrobacterium tumefaciens is a soil-borne soil-borne gram-negative bacteria. Gram-negative bacteria. It is rod-shaped and it is motile, and it belongs to the bacterial family of Rhizobiaceae. So it is soil-borne, it is gram-negative, it is rod-shaped, and it is motile in nature. This Agrobacterium tumefaciens. Now, Agrobacterium tumefaciens is actually a phytopathogen. Phytopathogen. And it is treated as nature's most effective genetic engineer. Nature's most effective genetic engineer. So some workers, they also consider this bacterium as natural expert of inter-kingdom gene transfer. So can perform inter-kingdom gene transfer. Can perform inter-kingdom gene transfer. So in fact, the major credit for the development of plant transformation techniques goes to the natural unique capability of Agrobacterium tumefaciens. Thus, this bacterium is most beloved bacterium by the plant biotechnologists. They love this bacteria. Now, there are two species of Agrobacterium. Basically, this Agrobacterium species. So, first is Agrobacterium tumefaciens, and second one is Agrobacterium rhizogenus. So, Agrobacterium tumefaciens is responsible of causing one certain kind of disease, and Agrobacterium rhizogenus is responsible for causing a certain kind of disease. So, Agrobacterium tumefaciens, it induces crown gall disease, and Agrobacterium rhizogenus, it causes hairy root disease. So, Agrobacterium tumefaciens and Agrobacterium rhizogenus. Now we'll be more over talking about Agrobacterium tumefaciens, and it causes crown gall disease. So we'll talk about crown gall disease. Crown gall disease, and the plasmid which is responsible for this is TI plasmid. So crown gall disease and TI plasmid. So Agrobacterium tumefaciens, how does it does this? So Agrobacterium tumefaciens, what it does, it infects the wounded and damaged plant tissues already which is damaged somewhere, infects damaged wounded tissues, and it leads to plant tumor, leads to plant tumor, and this plant tumor is called as crown gall. It's called as crown gall. So Agrobacterium tumefaciens is responsible to cause infection in already damaged and wounded plants. So the entry of bacterium into the plant tissue is actually facilitated by release of certain compounds from the plants. So the compounds which plants release, compounds which facilitates release of the compounds which facilitates infection of, which facilitates infection of bacteria are. And what are these compounds? These compounds are phenolic in nature. They are phenolic compounds. Phenolic compounds. These phenolic compounds can be acetoserine, it can be hydroxyacetoserine, and these are released by the wounded parts of the plants. The plant where it is in having certain kind of wound, from there it is released. So crown gall formation occurs when the bacterium releases TI plasmid into the plant cell cytoplasm. So for example, there is a branch and there is a cut, so there will be release of phenolic compounds. These phenolic compounds are going to help the bacteria enter into the plant, and when it will enter into the plant, what it will do, it will insert its TI plasmid into the plant cytoplasm, and then further the infection will occur. So a fragment of TI plasmid enters, a fragment of TI plasmid, fragment of TI plasmid enters the plant, and this segment of the TI plasmid is called as tDNA. So this tDNA gets integrated with plants plant cell chromosome, that is to the in the host genome. Thus, ground gall disease is a naturally evolved genetic engineering process. It's a natural process, right? So the DNA, the tDNA, specifically this tDNA, this we are saying this specifically tDNA, it carries genes that codes for certain kind of proteins which are involved in biosynthesis of growth hormones. This contains genes for auxin and cytokinin. Auxin and cytokinin. Shona is writing, hello ma'am, I'm watching your videos every day. Thanks for teaching in most simple and clear way. Thank you, dear. So tDNA contains the genes for auxins and cytokinin, and it also contains gene for opines. These opines are actually amino acid derivatives, and they are the sugar derivatives. So opines and agropines, these are the two things which agropines, these are the two things other than the genes for auxin and cytokinin which tDNA actually carries. So the growth hormones causes the plant cells. The role of growth hormone is to cause the plant cell to proliferate and to form the gall. So growth hormone causes proliferation of plant cells and causes gall. And the opines and agropines, they are responsible for being utilized as the energy carbon source for the bacteria, or they are the energy source in bacteria. So growth hormones and opines. So in such case, the opines and agropines are not normally a part of plant metabolism. Plant does not produce naturally opines and agropines. So the Agrobacterium tumefaciens genetically transforms the plant cells and create a biosynthetic machinery in the plant cell to produce the nutrients for its own. So as the bacteria, it multiplies, it causes infection. We'll write the flow chart. Bacteria multiplies. Bacteria multiplies and continue infection, and crown gall develops. Crown gall develops, and this is a visible mass of accumulated bacteria and plant material. It is, crown gall is made up of bacteria plus plant material. So crown gall formation is the consequence of the transfer, integration and expression of tDNA in the plant, in the infected plant. The genetic transformation leads to the formation of crown gall tumors which interfere with the normal growth of the plant. So there are many dicotyledons, many dicots which are affected by crown gall disease. So example of such are grapes, rose and stone fruit trees. So we'll write over here several dicots are affected by crown gall disease, So they are grapes, roses and stone fruit trees. Right, now we'll talk about a little bit about the tDNA or organization of actually Ti plasmid. Organization of Ti plasmid. So the Ti plasmid is approximately 200 kb long and it exists as independent exist as independent independent replicating circular DNA molecule in Agrobacterium pumifation. So it's 200 kb long, it exists independently. The tDNA is the part of the Ti plasmid. The tDNA is variable in length. It can be from 12 kb to 24 kb. So the part which is actually transferred is actually variable. So this tDNA has variable length of around 12 to 24 kb, and this depends on the bacterial strain from which it has been taken or from where it has infected the plant. Now, Nopaline strain of Ti plasmid have one DNA length of 20 kb, while the Octopine strain have the two tDNA regions, that is TL and TR, so that are the respectively 14 and 7.1 kb in length, right? So it is variable from the different strain. Now let us see a diagrammatic representation of Ti plasmid in a well depicted manner. So let us draw a Ti plasmid. This is our, I should have taken another page anyways. So there is a part for auxin synthesis, then there is a part for cytokinin synthesis, then there is a part for opine synthesis, then there is a part for opine synthesis, then there is this border region. This is the right border from where it will be cut. So this is the right border and this is the left border. This is 24 base pair repeat and this is also 24 base pair repeat. Now the other regions which are actually responsible for infection and all. So there is a virulence region. This region is virulence region or vir region. So vir region will contain vir genes. Then there is a replicator, that is the ori gene, from where the replication will begin. And then there will be a portion which is responsible for opine catabolism. These are the regions which are present in the Ti plasmid. This is the Ti plasmid of Agrobacterium tumefaciens, which is not completely transferred, but a part of it is transferred, right? We have already talked about that. Now let us talk about this tDNA part. This part is still here, it is tDNA, right? So we'll talk about tDNA. So this region has the genes for the biosynthesis of auxin. Then it has gene for the biosynthesis of cytokinin, and then it has got the gene for opine. These three are the things for the tDNA, and it is flanked by left and right borders, and these genes, that is the auxin, cytokinin O and opine, these are referred as oncogenes. These three genes are referred as oncogenes. Oncogenes, as they are the determinants for the tumor in the phenotype. They are responsible for causing the tumor. Responsible for tumor. So they are called as oncogene, right? After this, the tDNA borders, that is the left border and the right border. So this is the left border and this is the right border. So there is a set of 24 base pair sequence which is present on either side, that is the right and the left side of tDNA, and they are responsible for transferring the plant cells. And now it is moreover an established thing that right border is more critical for tDNA transfer and tumorogenesis. Right border is more important as compared to left border, right? So we have talked about the tDNA. Now let us talk about the vir region or the virulence region. So the genes which are responsible for the transfer of tDNA into the host plants, they are located outside the tDNA. They are away from the tDNA, and this region is referred to as the vir region or virulence region. So the region, the another important thing about this is it contains total 9 vir operons. So 9 different types of operons are involved which are responsible for the transfer of whole tDNA into the plants. So these 9 vir genes, they are actually like this: A, or I'll write vir A, G, B1, C1, D1, D2, D4, E1 and E2. These are 1, 2, 3, 4, 5, 6, 7, 8 and 9. So these are the 9 virulence genes which are responsible for transfer of this tDNA into the plant. Now the part that is left is opine catabolism. So opine catabolism region, this region actually codes for the protein which are responsible for the uptake and metabolism of opine, because now they are producing opine as an energy source, so they must be able to catabolize. So opine catabolism region, so it is responsible for the metabolism of the opine which has been produced. So besides the above three regions, or the previously described three regions, that is virulence region, tDNA and opine catabolism region, there are other places also which are there in the Agrobacterium tumefaciens, the eye plasmid, but these are the most important ones. So we have seen. So this was about the organization of Ti plasmid in the topic indirect gene transfer, right? Now we'll be seeing that how tDNA is transferred and integrated. So tDNA transfer and integration. So this complex thing can also be called as mechanism of tDNA transfer. Mechanism of tDNA transfer. So the process of tDNA transfer and integration into the host plant genome is actually like this. First of all, the thing that will occur is signal signal induction signal induction to agrobacterium. Signal induction to agrobacterium. So let us just read each and everything as we are writing. So signal induction, this is the thing. So how is the signal is induced? So the wounded plants, they actually release certain chemicals. So what are those chemicals? They are phenolic compounds. Phenolic compounds. Wounded plant release. Wounded plant release phenolic compounds. So these are these phenolic compounds, and it also releases certain kind of sugars. So these signals are recognized by agrobacterium. These signals are recognized by agrobacterium, and the signal they induce is as a result of the biochemical events in the agrobacterium, and it ultimately leads to tDNA transfer. These signals are recognized by agrobacterium tumefaciens and helps in transfer of tDNA. This is the first step, that is the signal induction. Now after signal induction, that signal is there that yes, now the plant is injured. The signal that is the most important signal is that the plant is injured. So now after the signal, the agrobacterium will perform its next thing. So let us see that what is the second step of this process. So second step is attachment of agrobacterium to plant. This attachment of agrobacterium to plant. Now in this process, the agrobacterium attaches to the plant cells through polysaccharides. Attaches to plant through polysaccharides. And these polysaccharides, specifically cellulose, is used for the attachment. And this cellulose is produced by actually bacteria. Cellulose from bacteria is responsible for attachment. Now several chromosomal virulence genes will be responsible for cutting and then transfer of tDNA into the plant cells, right? So attachment of agrobacterium to plant occurs with the help of a polysaccharide, that is cellulose, and which is produced by bacteria. Now production, another is production of virulence proteins. Production of virulence proteins. So how does this occur? Let us see this. So as the signal induction occurs in the agrobacterium cells, the agrobacterium cells, they attach to the plant cells, and a series of events are going to occur resulting in the production of the virulence proteins. And to start the signal induction by phenolics, the first gene which is stimulated is Vir-A. First, Vir-A is stimulated. Vir-A is stimulated, and this Vir-A in turn stimulates this Vir-A stimulates Vir-G. So the stimulation of Vir-A actually occurred due to what? Due to the phenolics. Phenolics stimulated Vir-A. Vir A stimulated Vir G. So this induces the expression of the genes of the Ti plasmid to produce the corresponding virulence protein, that is D1, D2, E2 and further. And certain sugars that induce the virulence genes have been identified. These are also produced. So the next step is production of tDNA strand. Production of tDNA strand. Now, the right and the left borders of tDNA, they'll be recognized by the Vir D1 and Vir D2. We shall write this, that right and left borders recognized by Vir D1 and D2. These proteins are involved in production of single-stranded DNA. These proteins will lead to production of single-stranded tDNA. Single-stranded tDNA. And its protection, because it's a single-stranded DNA, it cannot be transferred smoothly. It has to be protected. So its protection and export to the plant cell should occur. So this single-stranded tDNA gets attached with Vir D2. The single-stranded tDNA will be attached to Vir D2 protein. So this is kind of protection. After this, the fifth step is transfer of tDNA out of Agrobacterium. Transfer of tDNA out of Agrobacterium. How this is going to happen? Let us see this. That the single-stranded tDNA is actually in association of Vir D2 and is in complex association with Vir G, which is responsible for export of the tDNA outside the Agrobacterium. Responsible for export of tDNA from bacteria. Now, we have seen Vir D, Vir D1 and Vir G. There is another virulence gene which will play the role, that is Vir B. So the product of Vir B or the Vir B proteins will be responsible to form the transport apparatus. Transport apparatus. Vir B is responsible for formation of transport apparatus. So I'll just highlight that we have seen Vir D2, that is the protection, G is for export and B is for forming the transport apparatus. Right? It will form channel and those all things. Now lastly, next step is transfer of tDNA into plant cells and its integration. Transfer of tDNA into plant cells and integration. Transfer of tDNA to plant cells and integration. This we are going to. So let us take another color. Now what will happen, that we have seen that D2 is protecting, G has been in the process of export and B is for forming the transport apparatus. So tDNA will be in association with Vir D2 products And tDNA with D2 complex is going to cross the plasma membrane. This is going to cross the plasma membrane. Cross the plasma membrane. Now in the plant cells, the tDNA gets covered with the Vir-E2. So after this, it crossed the plasma membrane. In plant cells, what is going to happen? In plant cells, tDNA covered with Vir-E2. So this covering protects the tDNA from degradation by the nucleases. So E2 saves from degradation by nucleases. Saves from degradation by nucleases. Right? This is the thing. Now, Vir D2 and the covering which is protecting the tDNA from the degradation. So, Vir D2 and Vir E2. We have seen that D2 is also there. So, Vir D2 and Vir E2, they are going to interact with a variety of plant proteins which influences the tDNA transport and its integration. So, D2 and E2 are going to interact with various plant proteins for what things? For transport and integration of tDNA. For transport and integration of tDNA. This is the function of D2 and E2. Right? Now the tDNA transport is going to occur. So the tDNA, we'll write the complex, that is tDNA is with Vir D2 and Vir E2. So the plant protein complex enters the nucleus. This thing enters nucleus. The tDNA gets integrated into the plant chromosome. tDNA gets integrated in plant chromosome through a process which is called as illegitimate recombination. Through a process called as illegitimate recombination. So it is called as recombination. So this is different from homologous recombination. It is itself illegitimate recombination, and it does not depend on the sequence similarity. Most important thing, if we are saying illegitimate recombination, why we are calling so? Because it's not dependent on sequence similarity. Right? So we have seen the whole process of tDNA transfer, isn't it? Now we need to see that if we are using it for the gene transfer, how is it used? So let us go back to the place. Yeah, this place. So here, if in place of opine, cytokinin and auxin, if we replace this place with the gene of interest, the gene which we want to transfer, so the whole process can remain same, and just in this place the gene of interest can be put and it can be transferred into the plant. So that can be the indirect method of gene transfer. So this is the very first method which we have seen in today's class in the category of indirect gene transfer. So the tDNA can be manipulated, it can be used for the transfer of the gene of interest which we want to transfer, right? So this is the thing. about the indirect gene transfer method and through agrobacterium mediated. Now I told you that in indirect gene transfer also we have got two types. So other is virus mediated. Virus mediated. Virus mediated gene transfer. This is the second thing that we are going to see. So when virus, they are used as vectors, it is called as virus mediated gene transfer. So plant viruses, they are considered as efficient gene transfer agents. So virus mediated method is an efficient method. Efficient method. They can infect the intact plants and they can amplify the transferred genes through viral genome replication. Efficient method. Other than this, the important thing is they can infect the intact plants, infect the intact plants, infect the intact plants and and amplify the transfer genes through viral genome replication. So infect the intact plant, amplify the transfer gene, and viruses are the natural vectors for the genetic engineering. So we'll write natural vectors. They can introduce the desirable gene into almost any of the plant cells since it is the viral infection and they are the most systemic. So can infect any part of the plant cells or any of the plant cells. Can infect any plant cell because viral infection is a systemic infection, right? Because it's a systemic infection. Now, another thing that we need to see with this is plant viruses are non-integrative vectors. Plant viruses are non-integrative vectors. Plant viruses are not integrative vectors. So the plant viruses, they do not integrate into the host genome in contrast to the vector-based tDNA. Like in the tDNA, we have seen that it gets integrated, but here it doesn't get integrated. The viral genomes are suitably modified by introducing the foreign genes. So these are the things, and we'll see this in a short. And the criteria of virus vector being used as gene transfer method, what all things it should have. The virus should be capable of spreading from cell to cell through plasmodesmata, should spread through plasmodesmata. Then another thing is the viral genome should be able to replicate in the absence of viral coat protein and spread from cell to cell. And this is desirable since the insertion of the foreign DNA will make the viral genome too big to be packed. Then the recombinant viral genome must elicit little or no disease symptom in the plants, must elicit no disease in plants, and it should have a broad host range. Broad host range. Now some of the examples, famous examples which you should know for the transfer. So first is cauliflower mosaic vector. Cauliflower, sorry, it's not cauliflower, it is polymovirus. Polymovirus as vector. Polymovirus as vector. So polymovirus as an vector, why it is used? Because it is circular, double stranded, spherical in shape. So circular, double stranded, spherical in shape. Cauliflower viruses are widely distributed and are responsible for a number of economically important diseases in various crops. Then this colimavirus group has around 15 viruses in its own category, and cauliflower mosaic virus is the most important for gene transfer. So cauliflower mosaic virus is the most important one. It has got 15 viruses, and cauliflower mosaic virus is the most important one. And there are other colimaviruses as well, and they can induce the incarnation, um, edged viruses, Dahlia mosaic virus, Mirabilis mosaic virus, and strawberry vein broadening banding virus. These all are also the viruses which can actually lead to the gene transfer. Now, cauliflower mosaic virus, it is the most important one. Just give me a second. Let me put some slides over here so that we can get the blank page and we can write the things. So just give me a second. Yes. So cauliflower mosaic virus we are discussing. So cauliflower mosaic virus can infect many plant members such as crucifers and the tura. Very the tura, etc. And it can be easily transferred even mechanically. And another attractive feature of cauliflower mosaic virus is that the infection is systemic, and viral infection are mostly systemic, and large quantities of viruses are found in the infected cells. Large quantity of viruses found in infected cells. So this is the thing, and it is 8 kb long, and it is a relaxed but tightly packed circular DNA with 6 major and 2 minor coding regions. So the genes are from gene 2 and gene 7. They are not responsible for the viral infection. Total there are 6 major genes, and there are 2 minor genes. So amongst them, gene 2 and gene 7, they are not essential for the viral infection. And uses of cauliflower mosaic virus is for appropriate transfer of DNA into the plant cells, and does not contain any non-coding region where the foreign DNA does not can be inserted. It is fortunate that there are 2 genes which are of no use, that is gene 2 and gene 7. So these are the places where the foreign DNA can be inserted. Foreign DNA can be inserted. So it is therefore possible to replace any one of them and insert the desired gene, whichever we want, right? So this was about cauliflower mosaic virus. Another virus which is used for the gene transfer in plants is Gemini virus. Gemini virus. Then Gemini virus as vectors. Then RNA plant viruses. RNA plant viruses. So there are various ways of indirect gene transfer, but there are certain limitations which are actually associated with viral vectors as gene transfer. So limitations of viral vectors. What are these limitations? So the gene transfer is to transcribe the desired gene to the subsequent generation, that it should be further moved on. With virus vectors, it is not possible unless the virus is transmitted in the genes. So no surety of passing gene to next generation. Next generation. With virus vector, this is not possible unless the virus is seed transmitted. So, however, in the case of vegetatively propagated plants, transmission of desired trait can be transmitted to the next generation. Even in these plants, there is always a risk of losing the gene any time. So, it is not a very 100% sure way to transfer the gene for the next generation. Now we'll be talking about direct gene transfer. Direct gene transfer. Direct gene transfer methods. So in direct gene transfer methods, we are going to read what all things. Let me see. So the term direct or vectorless gene transfer is for introducing the foreign DNA directly into the plant genome. Introducing introducing foreign gene directly into plant genome. Plant genome. So direct DNA transfer methods, they rely on the delivery of naked DNA into the plant cells. Plant cells. So this is in contrast to the Agrobacterium or vector mediated DNA transfer, which may be regarded as an indirect method of transfer. So majority of the DNA transfer methods are simple and effective, and in fact several transgenic plants have been developed by this process. There are various limitations which we would discuss in the starting only. That what are these limitations? So limitations are, the major disadvantage of direct gene transfer is that the frequency of transgene rearrangement is high, and this results in the higher transgene copy number and high frequency of gene silencing. So I'll write over here high transgene, high transgene copy number. Shakira is saying hi ma'am, hello. High transgene copy number and high frequency of gene silencing. So I would like to tell that my mom is also attending this class presently. So hello mom, I hope you are understanding what I'm trying to convey. So my mom is also from biology background, so I'm sure that she must be understanding. So yeah, that this revision is going on for you mom by your child. So high frequency of gene silencing, then types of direct DNA transfer. Let us see that. Types of direct DNA transfer. So first is physical gene transfer. Physical gene transfer methods. Then in physical gene transfer methods, what all things comes is electroporation, particle bombardment, particle bombardment, microinjection, liposome fusion, then silicon carbide fibers, on carbide fibers. So physical, then in direct gene transfer methods, there is this chemical. So chemical gene transfer methods. So the chemical gene transfer method occurs with the help of polyethylene glycol, then diethyl amino ethyl mediated, dextran mediated, dextran mediated, then it is calcium phosphate mediated, calcium phosphate precipitation, precipitation. So chemical methods, polyethylene glycol mediated, then diethyl amino ethyl mediated, dextran mediated, and calcium phosphate mediated. After that, it is DNA imbibition by cells. DNA imbibition by cells, oblique tissues, oblique organs. Right, so the salient features of different methods for direct DNA transfer are many, which we are going to see that what are the salient features of direct DNA transfer methods, but that we are going to talk after a while. Now let us talk about the physical methods, some of the physical methods we need to talk. So we'll be talking about electroporation, we'll be talking about particle bombardment, microinjection, liposome mediated, and we have to do this. So let us start that. We are starting with electroporation. Electroporation. So electroporation basically involves the use of high field strength electric impulse. So electroporation, electric impulse to reversibly permeabilize the cells, reversibly permeabilize, reversibly permeabilize the cell membrane for the uptake of DNA, or uptake of DNA. So this technique can be used for the delivery of DNA into the intact plants or protoplast. So here what we can write, that intact plant and protoplasts can be transformed, can be transformed. Electroporation. So how this thing is done, I'll just tell you the outline because we are not going in deep into the detail. So the plant material will be incubated in a buffer solution containing the desired foreign DNA, and then it will be like there will be a vessel here, there will be buffer, there will be plant material, and there will be foreign DNA. Everything will be there, and this vessel will be subjected to voltage electric impulse, and this is going to result into the formation of pores. So the word itself is saying poration. So electricity and poration. With the help of electricity, pores are formed. So small pores will be formed in the plasma membrane, and through this pores the DNA is going to enter, and these DNA are going to integrate themselves with the host cell genome, and this is the way how it occurs, right? So what are the advantages of electroporation? This technique is simple, it is convenient, it is rapid, it is cost effective because we don't need any hi-fi costly chemical. We just need the electrical impulse and we need the buffer, which is available very easily. So the transformed cells are at the same physiological state after electroporation. They do not get changed, and efficiency of transformation can be improved by optimizing the electrical field strength and addition of spermidine. These are the advantages. There are some limitations as well. No, nothing comes with 100% benefit. So under the normal conditions, the amount of DNA delivered into the plant will be low. Efficiency of electroporation is highly variable depending on the plant material and the treatment conditions. And regeneration of plants is not very easy when they are uh, used as protoplast, right? So these are some of the things which are associated with electroporation. Second is particle bombardment method. Particle bombardment, and this particle bombardment is also called as biolistics. So in this, it is also called as micro projectile bombardment. Instead of particle, it can also be called as micro projectile. And this is the most effective method of gene transfer. We should mention over here that this is the most effective method of gene transfer, most effective method, and it is highly used for formation of transgenic plant. And this method is versatile, used all over the world. It is used for even mammalian cells, it is even used for microbial cells, and it is used for plant cells. So it is a versatile method. The micro projectile bombardment method was initially named as biolistic. Here we have seen that it is called as biolistic as well. So this word biolistic was actually given by its inventor, uh, Sanford in year 1988. So there are other names also for this thing. There are various names, so names should not be forgotten. So the names which are used for this is particle gun method, or it is also called as gene gun, and it is also called as blaster. These are the things which are associated with this. So what happens is in this a micro carrier will be used. So sometimes tungsten or gold particles they can be used, and these gold particles or tungsten particles will be coated with DNA, can be coated with DNA, and they are carried by macro carriers, and these macro carriers are inserted into a apparatus and pushed downwards by rupturing the disc, and the stopping plate will not allow the movement of macro carrier, while the micro carrier will be propelled inside the cell at high speed, and the DNA segments will be released and enter the plant cell and are going to integrate with the genome. This is the way how particle bombardment process is performed. After this, let us talk about microinjection. Microinjection. So microinjection is a direct physical method. So direct physical method. Direct physical method. Then it involves the mechanical insertion of desirable DNA into target cell. Direct physical method, mechanical, directly inside the cell. So this target cell can be or may be the one identified from the intact cell protoplast. It can be callus, it can be embryos, it can be meristem. So what all things can be used in this? Intact cells can be used. Intact cells can be used. Protoplasts can be used. Protoplasts can be used. Then callus can be used. Embryos can be used. Meristems can be used. So microinjection is used for the transfer of the cellular organelles and for the manipulation of chromosome as well. So how it will be done? The cell will be over here, and it will be actually held by the oil suction. There will be a suction, vacuum suction, oil suction, or any holding pipette. This is the cell, this is the nucleus, and this is the micro manipulator needle. And here also there will be pressure oil, and this is the injection. And here in this injection there will be filled DNA, so it will be manipulated this way. So the process of microinjection is complete through the process that the transfer of gene through the micropipette will occur into the cytoplasm or nucleus of a plant cell or protoplast. While the gene transfer is done, the recipient cells are kept immobilized in the agarose embedding and held by a suction holding pipette. The major limitation of microinjection is that it is slow, it is expensive, and it has to be performed by trained and skilled persons. These all are the limitations. After that, it is liposome mediated transformation. It is liposome mediated transformation. So we know what is a liposome. So liposome, in this case, liposome are artificially created lipid vesicles containing the phospholipid membrane, lipid vesicles, lipid membrane, phospholipid phospholipid membrane. So they are successfully used in mammalian cells for the delivery of proteins, for the delivery of drugs, etc. So liposomes carrying the genes can be employed to fuse with the protoplast and transfer the gene. So the efficiency of transformation increases when the process is carried out in the conjunction with polyethylene glycol. So liposome, how it will be done that a liposome with DNA material will be actually mixed in a solution where there will be cells also along with the DNA, and it will be left like that, and polyethylene glycol will also be added so that the efficiency increases. So addition of the protoplast to the surface occurs and its fusion at the site of attachments occur, and the release of plasmid occurs inside the cells, right? So this is the thing. So what are the advantages of this process? That being present in an encapsulated form of liposome, DNA is protected from the environmental degradations and damages. Then DNA is stable and can be stored for some time in the liposome uh, entirety, and it is applicable to a wide range of plant cells, and there is good reproducibility also. Now what is the limitation? So the major problem with liposome mediated transformation is the difficulty associated with the regeneration of the plants. This is the thing. Now silicon carbide fiber mediate transformation, we'll be seeing silicon carbide fiber mediated transformation. So in this the silicon carbide fibers are about zero point 0.3 to 0.6 micron meter in diameter, and they are 10 to 100 micron meter in length. So these fibers are capable of penetrating the cell wall and plasma membrane, and thus they can deliver the DNA into the cells. Silicon carbide fibers. So the DNA is coated. The DNA coated silicon carbide fibers are vortex with the plant material. During the mixing, the DNA adhering to the fibers enter the cells and gets stably integrated with the host genome. The silicon carbide fibers with the trade name Whiskers are available in the market. So it is available by the name Whiskers. What are the advantages of silicon carbide mediate transformation is direct delivery of DNA into intact walled cells and avoids the protoplast isolation. Procedure is simple, and the equipments are simple, so it is one of the methods of choice. Now coming to the chemical, this that was physical, now chemical gene transfer method. Chemical gene transfer method. So chemical gene transfer methods, first of all, is polyethylene glycol mediated. Polyethylene glycol mediated in the presence of divalent cations, that is calcium ions, is the uh, method of choice. So the polyethylene glycol and divalent ion is going to destabilize the plasma membrane of protoplast and render it permeable to the naked DNA and permeable to naked DNA. This way the DNA enters inside the nucleus of the protoplast and gets integrated. DNA enters nucleus. So the process involves the isolation of protoplasts. We have seen in yesterday's class that how the protoplasts are isolated, and their suspension and their adhesion, and then the addition of the plasmid, and how all these things are going to occur, that we have seen. So this method is quite a method of reasonable choice, and the advantages are a large number of protoplasts can be simultaneously transformed, and the technique can be successfully used for wide range of plant species. Then the limitations of polyethylene glycol is DNA is susceptible for degradation and rearrangement. So random integration of foreign DNA can occur, and regeneration of plants from the transformed cells is a little problem, right? So that is the thing. Now let us move towards this is the first method, that is polyethylene glycol mediated. Then the second method is DEAE, dextran mediated, DEAE dextran mediated method. So in this method, the desirable DNA can be complexed with the high molecular weight DE, DAE, that is diethyl amino ethyl dextran, and transferred. The major limitation of this approach is that it does not yield stable transformed cells. Then it is calcium phosphate co-precipitation method. And in this the DNA is allowed to mix with calcium chloride solution and isotonic phosphate buffer to form DNA calcium phosphate precipitate. When the actively dividing cells in the culture are exposed to the precipitation for several hours, the cells get transformed. And the success of this method is dependent on the high concentration of DNA and the protection of the complex precipitate. So addition of dimethyl sulfoxide actually increases the transformation efficiency. This transformation efficiency. Now, DNA imbibition by cells. Imbibition by cells and shoots. So some workers, they have seriously tried to transform the cells by incubating the cell suspension and tissue embryo and even seeds with DNA. So the belief is that the cell will get imbibed on it, the DNA will get imbibed on its own and the cells get transformed. But it is not a at all sure process that it is going to happen. It is just a matter of chance, and the probability is quite less for this thing. So like this, we have covered most of the thing in this category. Now let us talk about uh, just let me check the chats what you people have written over here. Let me just check that. Just a second. Uh, Okay, so I can see some of the students, they are writing some of the things. So, okay, now I think this is it for the class. We have tried to cover all the topics which are related to this thing. And lastly, as we say, let's crack CSIR UGC NET together. If you haven't subscribed our channel yet, please subscribe our channel for getting the notifications. Hit the bell icon and use my referral code FB01 for getting a 10% discount. Do not forget to use my referral code, and just hurry up in getting the subscription, because soon the prices are going to increase. Recently a major discount is going on from 31st of December. The prices are going to increase, so just do fast and try to get the subscription. And if you really like my video or you have gained something, give it a thumbs up. Thank you very much for your patient listening. We'll be doing some other interesting topic related to GATE syllabus in tomorrow's class, and we'll be meeting tomorrow morning 10:30 for CSIR, and in the evening for GATE. So just be there, be attentive, and try to. So this is it for now. We'll be meeting again tomorrow. If you have any doubt or query, or you want a specific topic to be explained by me, just write in the comment box. I'll be very much happy to take up your query. So it's a thank you from my side. We'll be meeting tomorrow again. So it's a final bye.